something stupid, Burgess.
On that test? Aren't you serious? What the heck is DNA replication? I just drew a straight line. That's what I thought it was. How deep and elaborate is DNA replication? Like, what's going on down there? Someone explain to me.
DNA is a molecule made up of two strands twisted around in a double helix shape. It carries genetic information necessary for life and is located in the nucleus of the cell. DNA is a polymer made up of monomers called nucleic acids, which are linked together in long chains. Each strand of DNA is made up of a sequence of 4 nitrogenous bases represented by the letters A, C, G, T. However, the two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the opposite strand. One strand has a 5' end and a 3' end, and because the other strand is complementary to the other strand, it has a 3' to a 5' end. Thus, the two strands run in opposite directions and are anti-parallel. DNA replication is important because without it, cell division cannot occur. That is, either one cell would get all the DNA and the other cell would get none of the DNA.
The first step in DNA replication is separating the two strands. This unzipping is done by an enzyme called helicase, which is represented by the pickaxe. The helicase cuts through the hydrogen bonds and divides the DNA strand into a leading and lagging strand. Single-strand binding proteins come in through the process and help the now divided strands stay as far away from other as possible. The result of the breaking of the DNA is a replication fork, and the beginning of the replication fork marks the start for the origin of replication. The old strand that goes 5' to 3' from left to right is the leading strand. Now, an enzyme called primase starts a process of replication in the leading strand. This enzyme makes a piece of RNA called RNA primer, and putting the RNA primer starts the process of making of the new strand of DNA. DNA polymerase then comes into the process and binds to the primer and will make the new strands of DNA. DNA polymerase can only add DNA bases in one direction, from 3' to 5' from left to right. One of the new strands of DNA, the leading strand, is made continuously, like shown.
The lagging strand, flowing from 3' to 5' from left to right, however, is anti-parallel and goes opposite directions compared to the leading strand. After the RNA primer is added, the DNA polymerase adds small pieces of DNA bases called Okazaki fragments. After each Okazaki fragment, a RNA primer is added and another Okazaki fragment is added. After this process, DNA polymerase comes in and takes out the RNA primers. Finally, the enzyme ligase comes in and pours in the necessary DNA pieces and completes the bond between all the DNA bases of the new strand. The process is semi-conservative because the new strand is made up of one old strand and one new strand.
Telomerase helps unwind the DNA strand in the beginning and helps wind the newly formed DNA strands into a double helix form again at the end.
Good morning, Mr.
Good morning.
Good morning.
Good morning. Good morning. So how do you think you guys did in the exam?
Did it pretty solid. Just studying amongst Twin and Savage.
Did you use any resources?
Yeah, we did, right? Yeah, it's just a YouTube video, DNA replication.
Did you use that video? No, but probably did still.
Yeah, it was by me. It was by me too. It was me, Luke, and Soka.
Okay. Well, you both watched the video and you both got an A+.
I like it.
You got me.
Me too?
No, you got an F because you didn't watch the video.
Oh,
Bro.
that's tough, man.
It's a good video.
Maybe I should check it out.
Watch it, please.
I will.